Good afternoon, ladies and gentlemen, and I'm Yayun Wang from National Taiwan University. And I'm very happy to be here sharing my study with you. And my research focuses on to identify the physiological functions of the Rhabdopsis MPF family transporters in responding to stresses. So what are the MPF transporters? MPF stands for nitrate transporter 1 peptide transporter family, and it has multiple members from the moths. So in Rhabdopsis, there are 53 genes, and in rice, there are 93 genes. So from the decade study, we have known that these genes have differential expression patterns. Okay, you may see here, this is the summary figure, show that the function of the nitrate transporters in Arabidopsis. You can see those genes has differential expression in roots, leaves, and flowers, and in the embryos. So they have different functions. For example, uptake the nitrate from the roots, transport the nitrate to the shoots, or remobilization of the nitrate between leaves. And more studies have shown that this family have unspecific specificity in the substrates. Okay, besides the nitrate and the dipeptide, they also can transport ABA, auxin, JA, and GA, and the glucosinolates. So we can help wondering why do plants need or have so many MPF transporters, and how do MPF transporters mediate the movement of diverse substrates? And some of the transporters can transport 2 or even more substrates. So does the co-transportation of these diverse substrates have physiological meanings? And yes, and they have some evidence shows that there are interplay between nitrate and hormones. For example, the MPF 6.3, or we call it CHL1, COL1, it can transport nitrate and auxin. And since the auxin transport is inhibited by nitrate, this gene will modulate the auxin level in the lateral root to influence the lateral root development. And another example is the MPF 3.1. It can transport nitrate and GA, and also show that this gene plays a role in hypocotyl elongation under low nitrate conditions. So my research are interested in how about these genes functions in stress conditions. So I focus on one particular gene, glucosinolate transporter 1, GTR1, or MPF 2.10. And it has shown that this protein is located to the plasma membrane and expressed in the vascular tissues. And it can mediate the multiple substrates, such as JA isoleucine, glucosinolates, GA, and the nitrate. And its expression is highly induced by salt, drought, wounding, and bacterial infection, and methyl JA. So I'm interested in the salt stress responses. Does this gene play a role in salt response? Okay, this is the expression pattern from the EFP browser on the website. So you can see that the expression pattern is highly induced by salt, specifically in the roots. And also, it's specifically induced by methyl JA, not by other hormones. So how about JA and the salt? Previous studies have shown that JA participates in the salt-induced responses, and it will inhibit the primary root growth. growth. So from this figure, you can see that in the JA biosynthesis mutant or the JA signaling mutant, the reduction or the root can maintain higher ratio compared to the when the plants grow in the sodium condition, it can keep higher, it can grow longer in the sodium conditions. So it means that JA mediated the salt-induced root growth inhibition. So the author proposed a model that when plants treated with sodium chloride, it will increase the jasmonic acid in the roots, which will induce the JA signaling and repress the cell elongation. So the roots cannot grow. So my question or I'm wondering, does GTR1 also persist in the salt-induced JA signaling? So I would like to know where can I put GTR1 in this pathway. So before I start, I would like to briefly introduce the JA signaling in Arabidopsis. Okay, without JA, the transcription factor responsible for JA responsive gene will be repressed by JAS, MINJA, and TPL repressor complex. And when there is JA isoleucine, this is the bioactive form of JA. The JA isoleucine will function as a glue that will induce the interaction between the CoE1, this is the receptor of JA isoleucine, and the JAS. And the JAS then will be ubiquitinated and then degraded through the 26 proteasome. So the transcription factor will be activated, and the JA responsive gene will be turned on, which these genes are JAS1, JAS5, LOX3, and MYC2. So in the following talk, I will use these four genes as the marker for the JA signaling. So first, I would like to confirm the microarray data from the website. So I treat the plant with methyl JA and sodium chloride and ABA to see whether the expression pattern of GTR1 is changed. So here you can see that the GTR1 is highly induced by methyl JA, sodium chloride, but not by ABA. So it is consistent with the website data. And later, I would like to know whether this induction is dependent on the CoE1, the JA receptor. So I did the same experiments using the CoE1 mutants. So you can see in the wild type, these four genes, GTR1, JAS1, JAS5, and LOX3, are highly induced by salt treatment. But in the CoE1 mutant, the induction ratio is highly reduced, almost abolished. So the salt-induced expression is CoE1 dependent. So I think now I can put the GTR1 here. So when there's a salt stress, it will induce the JA signaling, and the JAR1 will convert JA to JA isoleucine and activate the JA signaling through the CoE1. So next, I'm wondering whether the GTR1 also will affect the JA responsive genes expression. So I did the experiments in the GTR1 mutants. Okay. So in the wild type, these genes are highly induced, but in the GTR1 mutant, the induction rate is partially reduced. So this data indicate that GTR1 mediated the salt-induced JA responses partially. So I can modify a little bit about this pathway. So under the salt stress, JA signaling is activated, and the GTR1 will be induced through COE1 dependent pathway. And also there's another GT1 independent pathway, because you can see that the reduction is not completely, it still have a little bit induction. So how about the root architecture? Does the GTR1 also affect the root growth? So I also checked the primary root growth under salt treatment and methyl JA treatment. Okay. So under the salt treatment, you can see that in the wild type, the reduction is about 40%, or yeah, is 6%. It's only retained 40%. Methyl JA treatments, mutants, the reduction is not so severe as the wild type. The growth of the primary root in the GTR1 is less sensitive to salt salinity and the methyl JA treatment. And I also found the lateral root number and the density is also reduced in this mutant. You can see here it's the GTR1 mutant and the JAR1 mutant. So the lateral root number and the density is reduced. So GTR1 mediated JA signaling regulates the lateral root growth under salt stresses. And in the previous study, they already know that salt signaling will activate JA and repress lateral root growth, but nothing was talked about the lateral root. So I would like, when I found these results, I would like to have the answer why or how the GTR1 have this function. And then, okay, so I can here. So this is true. They can influence the lateral root. So I found this article. This article, the authors use GWAS analysis using 347 Arabidopsis thaliana accessions. They treat these plants with salt and observe the lateral root number and analyze the gene expression. So they want to correlate with the gene expression and the lateral root number. And then they found that higher expression of CYP72B2 correlate with more lateral root under salt stress, but higher expression of HKT1 correlates with less lateral root under salt stress. So that's give me a hint. So how about these two expressions in GTR1 mutant? So, and then I just do the RT-PCR and check the expression pattern. So we can see here, the CYP72B2 in the mutant is not changed, is not affected. Okay, this one is the GTR1C is the complementary line. However, in the HKT1, the HKT1 expression is higher in the wild type, even in the absence of salt treatment. So that will make my story more complete. So in the GTR mutant, the high expression of HKT1 may be the result of the lower number of lateral root. So this is my final model. So salt stress will activate JA signaling through COE1 receptor, and the GTR1 will be induced, and then induced JA signaling pathway, but repress HKT1 expression. So this will affect the lateral root development. And in the previous study, they have shown that HKT1 is a sodium transporter. So it's trying to keep the sodium in the roots. So they propose that higher sodium in the root will induce ABA signaling, which will arrest the lateral root growth. Okay, so this can be explained, but now I don't know how the GTR1 affected the HKT1 expression, and is there any relationship between J and the HKT1? This will be my I will study later. So all of the study have been published in the Plant Science paper here. And I would like to thank these two persons that done all the job in this paper, and the funding from the Ministry of Science and Technology in Taiwan and National Taiwan University. And this is my current lab members. And thank you. I would like to take questions.